Okay. All right. All right, guys. So see where we're at here. And then, like I said, I will basically review chapters 5 through 8, which is pretty dense set of chapters. All right. Oh, I think there's one other thing I wanted to do before I go to the review as well. So what I will do, like I did last time, I'll go over the quizzes and the homework to kind of start the review. And then if you guys have other questions, or if there's kind of topics that sort of come up that I might elaborate on a little bit, then that's the way I'm going to do it. Okay. So obviously I'm not going to go through all the lecture. So that's the way I did it last time. And then just interrupt me if you have questions. So in terms of, so like I said, lab exam 1 will be open until the end of the day, 11:59 p.m. tonight. Make sure you complete that. And then exam 2, like I said, I'll have that up this afternoon. Okay. And that's going to be available from today, this afternoon, until the end of the day on Saturday, because then you're going to have another lab quiz, which is really just a lab quiz that will be due or will start on Sunday, essentially. Okay. So I just wanted to go over that just really quickly. In terms of that lab, it's this enzyme lab. Okay. So everyone should have handed in that enzyme activity lab report and watched my demo. The lab quiz is just going to be only on that lab. Okay. So as long as you listen to and watch my demo and completed that enzyme activity lab report, you'll be fine on that quiz. Okay. Everything's just coming straight from that, the background information that's in the PDF, right? Here, and then basically what you put in that lab report. The one thing I just wanted to do before I go through the review, guys, is I said on the video that I would go over how to make a graph in Excel if necessary. Nobody asked me, but I had this file already. So if you just look real quick, and I think I looked at a couple people who had handed it in already, and it looks like everyone was, we knew how to make the, I don't know why I just hit that. Probably shouldn't have done that. That's not what I wanted to do. Sorry. Let me fix that. Oh yeah. That was stupid, huh? All right. So if you guys look here, right? So essentially, I just want to make sure that, again, if you haven't hit in your lab report, that you make the graphs right to begin with, so that I don't have to then give you feedback and ask you to resubmit. So basically, you're going into Excel, right? I mean, this is the temperature versus bubble height from that enzyme lab. You just basically have temperature in one column, bubble height in another column, and I just made up these numbers. They're just arbitrary. And you highlight, right? And you basically just go to insert. And in this case, we're making a line graph, which I believe is there, right? You click it. Oops, that's the wrong one. Let's try that again. Got to make sure you choose the right graph. I don't know what this is doing here. Oh, I know why, probably. 1 second. I don't know why that's choosing that weird graph. Oh, I know why. You have to choose, yeah. You got to choose scatter, and then you have to connect those lines, right? So if you choose line graph that way, which that's why I kind of want to show you guys, it'll draw this weird thing where it gives you these 2 lines. You just want one. So you can see my points are here, and then you just need to go in and format and basically just tell it to connect the points, okay? And that's essentially all you need to do for that graph. I mean, does anyone have more questions about that? Because I can go through it a little bit more, but I figured let me just pull it up really quick. Does everyone knows how to make a graph in Excel? All right. So anyway, if you have trouble with that, let me know. And then everything should be labeled, right? The axes need to be labeled. There needs to be a title. This title is not a very good title, right? It should be a better title. All right. Okay, so Enzyme Lab was due at 11 o'clock today. We will have a quiz on that enzyme lab. Exam 2 will be up, which is chapters 5 through 8. Let's review a little bit for those chapters. And then remember, we're in the next set of chapters this week that we're covering chapter 9, 10, and 11. Chapter 9 is already up. I'll get 10 and 11 up as well, okay? And then for the next module, right, for next week, if you guys look at the schedule, it's only one chapter, chapter 12. So it kind of gives us a little bit of a chance to sort of catch up on things, all right? And also you don't really have much in the way of lab. You have that one enzyme lab that was done, right? You don't have a lab that's due next week. You'll just have, I'll add one more lab next week. So again, it gives you a little bit of time to catch up, okay? So there is no lab that's going to be due next Thursday. There will be one more lab that's due that following Thursday, all right? Which would be the week of 6/22, which is our last week of class. All right. All right, so I'm going to start off by going through the quizzes. So quizzes chapter 5, and then I'll go from there. Please interrupt if you have a question. Here's my chapter 5 quiz. I'll do the homework. I'll do the quiz and then I'll do the homework, okay? All right. Okay, so this is only 10 questions, right? It says in a blank solution, there is a higher concentration of solute outside of the cell compared to inside of the cell. So if there is a higher concentration of solute outside, that means there's a lower concentration of water, right? But if we're referring to these terms, hypertonic, hypotonic, or isotonic. But remember, the reason why I was mentioning water is because we're going to think about how a cell will behave placed in that environment, right? And then what we're thinking about is how water moves, right? Whether it moves into the cell or out of the cell. But anyway, a solution, right? If there's a higher concentration of solute outside the cell, that's hypertonic, okay? And so if you want to think about how an animal cell will behave in a hypertonic solution, right? If a red blood cell is placed in a hypertonic solution, we're going to have water moving out of the cell, right? And causing shrinkage, all right? So these all kind of work together. All right. All right, blank requires ATP. So which one of these mechanisms requires ATP? Facilitated transport, osmosis, diffusion, or active transport? The answer is active transport, okay? I mean, the active part of it suggests that energy is required. All the following are membrane proteins, except which one here? So it's translation proteins. So I just, right, so channel proteins, receptor proteins, carrier proteins, those are all proteins that sit within the plasma membrane. Translation proteins are just proteins, enzymes that are involved in the process of translation, which we haven't talked about yet. So like I said, interrupt me if you guys have a question. Blank is the diffusion of water across a selectively permeable membrane from high concentration to low, right? So diffusion of water, osmosis, okay? And again, particularly we're talking about the movement of water across a semi-permeable membrane, whether water is moving from outside the cell to inside the cell or inside the cell to outside the cell, right? And remember, the movement of water is going to change depending on what that extracellular environment is compared to the intracellular environment, okay? Remember, molecules have the tendency to move along their concentration gradient, okay? So for this, it's osmosis. All right, all the following are components of the cell membrane except what? So integral proteins, right, are proteins that span through the phospholipid bilayer. Phospholipids, obviously part of the membrane. Testosterone, probably not. Glycolipids, right? Remember, these are lipids that have carbohydrate chains on them. And then peripheral proteins are proteins that don't span through the membrane. They sort of sit, they're sort of indirectly associated with the membrane, and they sort of face towards the inside of the cell. And so the answer here is testosterone. What could we have sort of replaced testosterone with? Cholesterol, right? Cholesterol is part of the membrane. It's a steroid. It is involved in maintaining the fluidity of the membrane, okay? All right, blank proteins help the body recognize when it is being invaded by a pathogen so that an immune response can occur. These are cell recognition proteins, right? All right, during blank, a vesicle inside the cell fuses with the plasma membrane and releases its contents into the extracellular space. So either endocytosis or exocytosis. If we're talking about endo, right, that's taking something in. If we're talking about exocytosis, it's releasing, right? The cell is releasing something. And so the answer is here is exocytosis. All right, carbon dioxide and oxygen can readily diffuse across the plasma membrane. True or false? The answer is true. All right, blank proteins have a shape that allows only specific molecules to bind. The binding of this molecule then leads to a cellular response. Okay, so the answer there is receptor. And so that's kind of like, you know, the presentation, that assignment that we omitted, you know, kind of gets into more detail about what happens when a drug, in that case, we were going to focus on a drug, right? Like an opiate drug, binds to its receptor on the membrane, what then happens inside the cell, and like, where are those receptors, right? What cells have those receptors? So it was getting, just to kind of get a little bit more in depth into kind of the sort of receptor function. Okay, so that's sort of where that assignment was coming from. But like I said, we don't have enough time really to spend on that, so I don't want to just throw it out there. So anyway, last question. Water crosses the plasma membrane through a channel called an aquaporin. Okay, so that is a special channel that allows water to be transported across the plasma membrane. All right. Okay, so that's that for chapter 5. I'm going to go over the homework questions, and if anyone has any other questions, please let me know. So let me go back. All right, so let's look at these, the chapter 5 homework questions. Okay, so it says, explain the role of proteins in the fluid mosaic model of the plasma membrane. So, I mean, you can have, you know, a couple, you know, different answers here, and everyone would answer this in their own way, which is fine. That's what I want. But, you know, one, we talked about, or I talked about proteins, you know, being embedded in that plasma membrane, right? And basically, we listed a whole bunch of different functions of membrane proteins, right? Cell recognition, enzymatic proteins, receptor proteins. So basically, you sort of could list some of those different functions that we talked about. You could also talk about, you know, proteins being integral, right? Like spanning the plasma membrane, like I mentioned, and also peripheral proteins, okay? Those integral proteins, right? Like carrier proteins and channel proteins are integral proteins. As our receptor proteins, okay? So it was just basically getting at that, you know, the discussion or the sort of list of all these different functions of these membrane proteins. Any questions about that? We got that? So make sure you do know those, you know, the different functions of those proteins, okay? Number 2, explain the difference between passive and active transport across a plasma membrane and give an example. So if we're talking about passive transport, right, that means it does not require energy. And these are going to be molecules moving along their concentration gradient. I mean, you can even think about, you know, water moving along its concentration gradient. I mean, you could give any example of that. I mean, you could give an example of, even, I mean, technically passive transport, like facilitated transport is technically passive as well, right? So you could give an example of sodium ions moving through the sodium channel, right? Along their concentration gradient. Anything like that would suffice. In terms of active transport, right? This is when the cell's gonna be able to, it's gonna move molecules against their concentration gradients, right? So if you have a, you know, think about inside the cell versus outside the cell. If there's a low concentration here and a high concentration here, normally molecules would want to move that way, right? But it's going to be pumped against its gradient. And so we call it pump. When you hear the term pump, it sort of probably suggests energy, right, involved. And so you're always going to involve energy. So we talked about like the sodium potassium pump. That's an example that you could have given. And I explained that that's super important in maintaining membrane potential or the charge across a membrane, which is particularly important to muscle cells and nerve cells, right? Those cells that can conduct impulses, so, or generate action potentials. So anyway, that's an example of that. But it's more kind of just being able to explain what we mean by those two different processes. Does anyone have any other questions about that? Okay, so any questions about chapter 5? Okay, so we pretty much just reviewed chapter 5. Chapter 5 focused on the plasma membrane, right? And movement across the plasma membrane. We also hit a bunch of those topics. Oops. We hit a bunch of those topics in the lab, right? That diffusion and osmosis lab. We covered a lot of that stuff that's in chapter 5 in there as well. So you may, you sort of got a little bit of reinforcement there. So I don't know if anyone has any other questions about that. I have chapter 5 here if there's anything we needed to pull up, but so that was me. I was mentioning the sodium potassium pump, right? That was our example of active transport. Just want to see if there was anything else really. I think most of this is pretty straightforward. The beginning part really talks about those membrane proteins and those different functions. Also, in these slides, right, it also gives you an example of each one of these sort of proteins, or an example of what might happen when something goes wrong with these types of proteins. Okay, so that's definitely something to kind of just pay a little bit of attention to, just to remind you guys. I sort of reminded you of that during the recording, but I'll remind you again. The other thing is, in terms of this in terms of exocytosis and endocytosis, we should know what that is, right? This is then the 3 different types of endocytosis. Okay, so just be familiar with those. In terms of these junctions between cells, honestly, it's just knowing that, yeah, I gave you 3 different examples of different types of junctions. I don't really harp too much on this and ask you too much about it. Okay, so I said that in the recording too, just to remind you. So don't go crazy, studying all the differences, right? Like, I'm not going to ask you to identify the difference between these different types or anything like that. Let's just kind of just be familiar with the different types that exist, and that's pretty much it. Okay. All right. So I am going to move on to chapter 6, unless somebody, like I said, has a question. All right, so let's go over that chapter 6 quiz and then the chapter 6 homework, okay? So chapter 6 was sort of a setup. We talked a little bit about enzymes, right? Which then corresponds to that enzyme lab that was due today. And then it's sort of a setup to things that are happening in chapter 7 and chapter 8, all right? In terms of like oxidation and reduction and things like that. So let's look at this as well. Okay, in blank inhibition, the inhibitor binds to the enzyme in the active site. Okay, so in this case, the inhibitor, whatever that inhibitor is going to bind right into the active site so that then the substrate can't get there. Okay, and so we call this competitive inhibition. If it was non-competitive, the inhibitor would be binding to an allosteric site or a separate site, not at the active site. Okay, so that competitive inhibitor binds right to the active site, and then it keeps the substrate from binding. All right. So again, remember that enzymes, or it's important for the cell to regulate or be able to regulate the activity of enzymes. Okay. And so inhibition or enzyme inhibition is like a thing that happens a lot, right? It's a major regulatory mechanism that the cell uses. So if enzymes or a certain enzyme was just turned on all the time, it would keep catalyzing its specific reaction. And there's many times where the cell wants to turn that down or turn that up. Okay, so this is actually a really important sort of process that occurs in the cell. All right, so it's not just something that happens like sometimes, it's happening all the time, this regulation. All right, blank energy is stored energy. Kind of an easy, direct question, right? Potential energy. All right. All the following affect enzymatic speed or enzymatic activity, right? Except what? And these were things that we tested in that enzyme lab, right? So pH, temperature, substrate concentration, or the shape of the product? The answer is obviously shape of the product, okay? We know that temperature can affect enzyme activity. In fact, usually it sort of generally increases it until a point, right? Because at some point that enzyme becomes denatured and then the activity is completely gone, okay? And when you graft that, and you kind of saw that, and I showed you that sort of example when I was showing you that graph, but you could see that, right? You get a dramatic decrease. pH, right? Enzymes are going to work at an appropriate pH. If that pH is changed in any direction, it will affect the activity of the enzyme, okay? Substrate concentration, how much substrate there is going to also affect the activity. All right. Molecules that are required by enzymes to bind to the active site in order for the enzyme to be able to bind the substrate and work properly are called cofactors, right? So remember I mentioned sort of coenzymes, cofactors. Remember that many of your vitamins, right, actually are cofactors for enzymes, okay? And so if you're deficient in a vitamin, a certain vitamin, it will affect that particular enzyme to which it serves as a cofactor, okay? All right. In blank, the substrate is broken down into smaller products, okay? So here we're talking about degradation. And we were talking about, we've been kind of talking about degradation reactions since like chapter 3, right? Chapter 3 on, when we talked about biomolecules. And so you have a substrate, right? Think about like starch, and then it being broken down into glucoses, right? Or smaller subunits, okay? So this all fits in, but now here we're focusing on the enzymes, okay? And bringing the enzymes into it. All right. As temperature increases, the activity of an enzyme increases, again, only to a certain point. Once that temperature increases high enough, it will then dramatically decrease. So, I mean, I probably should word that a little bit better or give you a little more clarity in there. But again, in general, temperature is going to increase the activity of the enzyme just because molecules are going to move around faster and they have more of a chance of interacting with one another. But again, once they reach a certain temperature, the enzyme will be denatured and will no longer function. All right, energy can be created and destroyed. The answer is false. It cannot be created or destroyed. That is that first law of thermodynamics, right? However, it changes forms, okay? And a lot of times, whenever you have sort of energy transfers, some of it is lost as heat, right? It's never completely transferred, okay? The molecule that binds to the active site of an enzyme is called, and I guess I've been using that term in a couple of questions already, substrate, right? The substrate binds to the active site, okay? And remember that enzymes are specific, right? They only bind specific substrates. And again, we saw that in the lab, right? In that enzyme lab, I think almost each one of those experiments, sort of, you had a tube where you didn't have the substrate or you didn't have the enzyme or something like that, right? And the reaction's not going to happen. So that enzyme can only catalyze one specific reaction. There are no general enzymes that just catalyze any reaction, right? They're super specific. All right, the conversion of ATP to ADP is a blank reaction. It releases energy. Okay, so it's exergonic, right? Exergonic reactions are going to release energy. Endergonic are going to require the input of energy. Okay, so obviously the conversion of ATP to ADP is a super important reaction, right? We say that ATP is the energy currency of the cell, right? So many reactions in the cell that are endergonic, right, or that require energy are coupled to the breakdown of ATP, okay, because it releases energy and then that energy is used to drive that other reaction. All right, last question. Blank refers to the loss of electrons or the loss of hydrogen atoms. So here we're talking about oxidation and reduction. Remember, there's a mnemonic oil rig. Oxidation is loss, reduction is gain. And I told you that it's best to kind of, for our purposes, to think about the movement of the hydrogen atoms, right? Rather than the electrons, because you can kind of see that when you look at the reaction. If we're talking about loss, right? Oxidation is loss, reduction is gain. So the answer here is oxidation. All right. Okay, so let me now go to the chapter 6 homework. Again, if you have questions, interrupt me. Where is my chapter 6 homework? Here it is. Okay, define potential and kinetic energy. Provide an example of a conversion from potential to kinetic energy. So again, you can have a couple different examples here. In fact, I like when people sort of come up with their own example, right? In terms of potential energy, it's stored energy, right? Kinetic energy is sort of that energy of motion, right? In terms of an example, I had this one, you know, the one picture that was in the lecture, I can kind of pull up just real quick. You know, it was the example of, that's the wrong, of the moose, I think, eating, right? I don't know, eating some plant and then it converting that, you know, into that sort of energy of motion, that kinetic energy. Where are we? Chapter 6, right? Right? So, it was kind of this picture, I guess, here is showing you this sort of flow of energy here, right? That there, you know, you have this sort of stored energy in the leaves of the plant. That moose eats those leaves, and then obviously, you know, that gets broken down and then ultimately into glucose. That glucose can then be converted into, you know, usable energy, right? And then this is showing you him moving is that mechanical or that kinetic energy. Okay, so I mean, you could have came up with a couple different answers there. That's sort of up to you, but just understand that we have the stored energy and then kinetic energy, which is that sort of energy of motion. Okay, and then let me just, for the next question, I guess I need this too, sorry. So for the next question, in terms of the ATP cycle, this is what I was referring to. So let me just pull everything up. Okay, it's to understand kind of, I just talked about this, I guess, before, but I talked about the breakdown of ATP to ADP, right? And that it's an exergonic reaction. This going from ADP to ATP is endergonic, equally as important, right? Because the cell needs to make sure it can regenerate that ATP to then use, you know, to be able to use this as an energy source, okay? So you can basically see the sort of breakdown of ATP, right? By cleaving off this one phosphate to ADP and then adding another phosphate back and going back to ATP. So that's all I meant by the cycle. It's just, you could have just described it as, you know, ATP being broken down into ADP, right? Which is exergonic and releases energy. And then ADP, you know, plus a phosphate, or ADP and then a phosphate being added to be converted back to ATP, which is endergonic, okay? So that's basically what I was expecting you to know. I mean, and you don't have to draw it out or anything like that, right? It's just knowing going from ATP to ADP, ADP back to ATP, okay? All right, so the last question I kind of already talked about too, competitive versus non-competitive inhibition. So here we're talking about enzymes, right? So again, let me just, I probably should just pull up a picture just to kind of review this with you. And there's actually one other thing that's related to this that I sort of want to review. Okay, so one thing here. So this is showing you the cofactors, right? So in this case, and this is sort of the opposite of the inhibition issue, right? The cofactor is going to bind, and that's going to facilitate the binding of the substrate, right? So that cofactor is necessary for the substrate to bind and basically is necessary for that enzyme to be active, right? But you have other types of molecules that act as inhibitors, right? And so in competitive, right, we said that the inhibitor will bind to the active site. So if you sort of, instead of this being a cofactor, this can be some sort of inhibitor that binds. And when that's bound, the substrate will not be able to bind. Okay, that's competitive. In non-competitive, the inhibitor would bind somewhere over here to some allosteric site. And I think I have a picture of that somewhere. Why do I not? I guess I don't really have a good picture there. I guess it's kind of here is the only picture I have. So let's go back to that. I guess I like this one better. right? In non-competitive, the inhibitor would bind somewhere else, some allosteric site, and then that would change the conformation of the enzyme so that then the substrate would not be able to bind, okay? So the inhibition, both competitive and non-competitive, sort of have the same end result in that the substrate can't bind, but the way that it happens is different, okay? Another thing to just mention, because I know this is on the exam, understanding this here, okay? So this is, it's kind of combining a couple things or a couple concepts, right? The concept of a metabolic pathway, right? Which I definitely talk about in the beginning of the lecture. In the fact that you have, right, here's your kind of your first substrate that you're starting with and your first reaction that's catalyzed by enzyme one, right? And you have a series of reactions all catalyzed by different enzymes to form an end product, okay? So that's that metabolic pathway. Now, what happens, again, I told you that the cell has to be able to regulate things all the time, right? So it has to have mechanisms to turn this pathway off once enough of F is produced, right? So when enough of it is produced, right, you can see here, it binds to the enzyme at an allosteric site, which is marked here. And what does it do? It changes the active site so that this substrate A can no longer bind, right? And so that's what this is showing you here. So again, you can think of this pathway as maybe the production of testosterone, okay? And there's a whole bunch of different reactions that occur to produce that end product testosterone. If there's enough testosterone around, cell needs to shut that down, right? So the testosterone itself will actually bind to that first enzyme in the pathway and inhibit it, so that none of these other reactions will then occur, okay? And so there are many, many pathways that exist this way in the cell and are sort of organized this way, okay? So make sure you guys understand this. It'll definitely be on the exam as an essay question, okay? Any questions about that, guys? All right, moving on to the last quiz is chapter 7-8 quiz and the chapter 7-8 homework, and then we are done. One other thing, just in case, I just remembered this is here. This is also, I think these equations come up again in the photosynthesis and the cellular respiration chapter, but I'm not sure. So anyway, you should be familiar with these is the point. That's why I made sure that they're in here. So in terms of this general equation for photosynthesis and the general equation for cellular respiration, I won't ask you to write it out yourself, right? But you should be able to recognize it, okay? And you can kind of see that they're opposites of each other. You also, and I point this out in the recorded lecture, should know, you know, if I ask you about something being oxidized or reduced in these reactions, we should know that as well, right? So if we look here in photosynthesis, carbon dioxide going to glucose here, right? Is carbon dioxide oxidized or reduced? Right here, what's happening is you're gaining hydrogens, right? Or gaining electrons. So reduction is gain. So carbon dioxide is reduced, okay? If we talk about water, going from water to oxygen, you're losing hydrogens or losing electrons, right? Water is oxidized, okay? So be able to recognize these general equations and know what's happening in terms of oxidation and reduction. All right. Okay, so like I said, let's just go through those last two chapters and we will be done. So sorry, I know this is long, but I think it's worth it to get a little bit of a review in. All right, so chapter 7-8 quiz first. And then I'll go back to the homework. Okay, so acetyl-CoA is produced during which phase of cellular respiration, right? The prep reaction, glycolysis, citric acid cycle, electron transport chain. The answer is the prep reaction. And remember, make sure we know where each one of these phases is taking place in the cell, right? Okay, a blank consists of a pigment complex and electron acceptor molecules within the thylakoid membrane. Okay, these are the photosystems. Remember, there's photosystem 1 and photosystem 2, and these are part of the light reactions of photosynthesis. All right, fermentation is an aerobic process, true or false? So what do we mean by aerobic, right? It requires oxygen. Okay, fermentation does not require oxygen. It's not aerobic, it is anaerobic. Okay, so the answer is false. Blank does not require oxygen. Glycolysis also does not require oxygen. Remember, you'll go through glycolysis, and then again, if there's no oxygen around, it can go one step further, right? I mean, even in certain cells in our body, this can happen in terms of the fermentation part, but also, you know, yeast, some bacteria, right, will go through fermentation and in the process produce things like alcohol, right, or lactic acid, and carbon dioxide. Okay, so again, remember, glycolysis does not require oxygen, and it occurs in the cytoplasm of the cell. All right, I just said that. Okay, so glycolysis, cytoplasm, does not require oxygen. All right, a net gain of blank ATP are produced following glycolysis. So it's a net gain of 2, right? Remember, you put 2 ATP in and you get 4 out. The net gain is 2. All right, in cellular respiration, glucose is blank and oxygen is what? So if we go back, right? Where was I? Chapter 6, right here. Okay, we go back to this general equation, and on the exam, I usually show you the equation. So if we're talking about glucose, right, going from glucose to carbon dioxide here, we're losing hydrogens, right? So glucose is oxidized, right, and oxygen is reduced, okay? So glucose is oxidized and oxygen is reduced. All right, organisms that produce their own food are called, this is an easy question, right? Autotrophs. So we're talking about plants that can produce their own carbohydrates, their own food. Autotrophs, heterotrophs cannot make their own food. They're going to generally ingest food that the autotrophs make. In photosynthesis, the carbon dioxide is blank and the water is what? We just did this already, right? So if I go back to that again, just to remind you, right? Carbon dioxide is gaining hydrogens or gaining electrons, okay? So carbon dioxide is reduced and water is oxidized, right? Carbon dioxide is reduced and water is oxidized. Hopefully I got that right. The light reactions of photosynthesis take place in the blank of the chloroplast, in the thylakoid membrane. The blank reactions of photosynthesis provide energy for the what reactions of photosynthesis? So the light reactions provide energy for the dark reactions, or Calvin cycle. A lot of people don't like to refer to them as the dark reactions. So light reactions provide the energy for the Calvin cycle. Remember, in the Calvin cycle is where the carbohydrates are made, right? And again, the light reactions are just providing the energy for that. In the blank step of the Calvin cycle, G3P is formed. Okay, so now if we go back to here, where are we? Chapter 7. I thought I had chapter 7 out. I do, it's over here. Okay, and we go back to, remember, we just want to kind of, I told you for photosynthesis, for the Calvin cycle, to know those 3 main phases, right? And what's happening at each one of those. So carbon dioxide fixation, carbon dioxide reduction, and then Ruby P regeneration. So in terms of kind of what's going in and sort of what's coming out at the end of each one of those phases, right? So we're talking about the formation of G3P, right? Oopsie, hello. And so that's here, right? Carbon dioxide reduction. Just to remind you of that. All right, so reduction of carbon dioxide. The complete breakdown of glucose in the presence of oxygen results in the production of how many ATP molecules, right? About 36 to 38. Okay, so this means going through all of those steps of cellular respiration, right? Starting from glycolysis and then ending with the electron transport chain and chemiosmosis. All right, in the non-cyclic pathway of photosynthesis, remember I told you this is what we focus on. We really didn't go through the cyclic pathway and the differences, but I just told you that I have to, tell you that it exists. Electrons move from photosystem 2 to photosystem 1, and the answer to that is true, okay? Hydrogen ions move from the thylakoid space to the stroma of the chloroplast through what? ATP synthase. And that's what then produces ATP. So remember, ATP synthase and the movement of hydrogen ions through ATP synthase, right? We call that chemiosmosis. That is going to happen in cellular respiration and also in photosynthesis. So make sure we sort of understand that process as well. All right. Okay, so let's go back. Last thing to do is just to go over the homework questions. I think they were pretty basic. I don't think I asked you too many questions, did I? Yeah. All right, so again, I told you guys those overview pictures, and I'll pull them up. Actually, I'll probably pull that up right now, okay? Because to be honest, all of these questions can just be answered by those overview pictures, both of photosynthesis and of cellular respiration, okay? So let me just do that real quick. And so that's that here. I guess that's that. I guess that's enough there. I just wanted it in the bit. Let me see if that's the sort of biggest. Yeah, I guess that's fine. Yeah. Okay. So here, right? If we're going to talk about the light reactions and the Calvin cycle, remember the light reactions, the function of the light reactions is to produce energy for the Calvin cycle. The function of the Calvin cycle is to make carbohydrates, right? And essentially, it's not even just making carbohydrates. Remember that G3P molecule that comes out of the Calvin cycle can be used to make, proteins and lipids as well, okay? So remember, I'll just remind you of that too, just so that we don't get confused, right? So that's this part here. Remember, what comes out of that Calvin cycle is G3P. But that G3P has the ability to basically form any molecule that the plant needs, okay? So like I said, again, I'll reiterate the point that this overview figure is probably the most important figure, right? Because again, it's showing you how the light reactions relate to the dark Calvin cycle, okay? Light reactions produce energy for the Calvin cycle. Calvin cycle makes food, makes carbohydrates, makes G3P. But essentially that's where the synthesis is going to occur. The other question asks about cellular respiration, right? So same thing, you can go to that overview figure, and then we shall end there. I probably can just look at it this way because it kind of has everything there, right? So it's this figure here where you can see here's glycolysis occurring in the cytoplasm, right? Showing you what's coming out of that, NADH, right? You get a little bit of ATP that's generated directly as well. Remember that the NADHs and the FADH2s, they all go to the electron transport chain and they act as electron carriers, right? And so super important because most of the ATP that's going to be generated is in the electron transport chain and chemiosmosis, okay? But again, if we go, so you can pretty much sort of describe each one of these phases by looking at this overview, okay? That prep reaction is going to form acetyl-CoA from pyruvate. That CoA is then going to enter into that citric acid cycle. And again, you're going to get ATP formed, right? But you're also going to get more NADHs and FADH2s, okay? The last part of this is this, okay? So let me just go over this and we're done with this. So this is the electron transport chain, right? Showing you the movement of electrons from, these protein complexes essentially that sit in the membrane, right? In the inner mitochondrial membrane. And then remember what happens is that you have hydrogen ions that are released and they're essentially pumped across here. And so all of these hydrogen ions accumulate out here, right? It sets up a steep gradient, and therefore these hydrogen ions will have the tendency to move along their gradient this way. The only way the hydrogen ions can go that way is through this ATP synthase, okay? When they move through the ATP synthase, ATP is generated. So that's where most of the ATP is generated. It's not really directly from the electron transport chain, right? But indirectly it is. Because of the electron transport chain, you get all of these hydrogen ions that accumulate here, the gradient set up, they then can move through that ATP synthase and ATP is generated, okay? So I think I pretty much kind of reviewed everything. Again, I know it's a lot. I mean, there's a lot of information in there. All right, guys, I don't know if there's any other questions. Oh, and I also, in number 3, asked you, again, you can see where carbon dioxide and where water are released through those steps as well. But, we mostly you want to focus on kind of what's going in and what's coming out of each one of those phases, how many ATPs are generated, and also how many NADHs and FADH2s are generated, right? And again, I like that overview figure because it shows you how all of those phases fit together, okay? Again, make sure you understand that electron transport chain and the chemiosmosis, the hydrogen ion gradient and all that, how that's set up, because that's the mechanism that generates all the ATP. So it's super important to understand that as much as you can. All right, guys, any other questions?
Could you just briefly go over the steps one more time? You don't have to pull up the figure if it's just how they all connect.
Yeah, so I mean, if we're thinking, so again, we always kind of think about, we think about starting with glucose, right? So which is fine, but remember too, at the end of the chapter, I kind of tell you that, you could start with breaking down fats and things like that, and they feed into different places. But let's just kind of talk about, so if you're starting with glucose, right? And you're starting with glucose in the cytoplasm, okay, glycolysis is going to happen, right? Glucose is going to be broken down into pyruvate, right? You have 2 molecules of pyruvate, and in the process, you get a little bit of ATP that's generated, okay? That pyruvate then enters into the mitochondria, and then the prep reaction occurs, right? And so pyruvate is converted into that acetyl-CoA, and essentially that acetyl part acts, it's sort of like a carrier, right? I can pull up the figure real quick though, just to show you. I'm gonna just go to that. I just want to go to that overview thing here, right? So that basically that acetyl group then enters into the citric acid cycle here, right? So that CoA kind of gets released. The acetyl group, right, which is these carbons, right, enter into this citric acid cycle. And then we go through that citric acid cycle. And as you move through that stepwise sort of series of reactions in the citric acid cycle, you get lots of NADHs and FADH2s that are formed, also ATP directly, right? Also, sorry, I kind of failed to mention at each one of these points, the glycolysis, right? You have NADH that's formed, right? Then prep reaction, more NADH. They all go to the electron transport chain, okay? And they basically are bringing those electrons to the electron transport chain, right? And then those electrons in the electron transport chain, which is now sitting in this inner membrane here, right? It's just a series of sort of protein complexes that sit there. And so those electrons get passed along. And as they get passed along, those hydrogen ions are released, right? And remember, those hydrogen ions accumulate, purposely, they accumulate here. Okay, this is in that space. So here, this is in the intermembrane space here, right? So if the electron transport chain is sitting here, these are accumulating kind of in this space between the outer and the inner membrane, okay? That sets up the steep gradient so that they then have this tendency to move this way. The only way they can move is through this ATP synthase. So each time the hydrogen ion moves through the ATP synthase, there's ATP that's generated, okay? And that's where most of the ATP is coming from. So you can kind of see how sort of each preceding phase is gonna produce the sort of substrate for that next phase, right? Does that make sense? Does that help?
Yes, thank you so much.
Sure. All right, any other questions, guys, or anything else you want me to review? Yeah, all right. All right, guys. So like I said, that exam 2 will go up hopefully sooner rather than later, because like I said, I have it ready to go. Try to import it like 3 times and there's issues every time. So hopefully I don't have to go one by one. And then just be on the lookout for the recordings for chapter 10 and 11. Those will go up in the next couple of days as well. Okay. And then remember, for next week, it's just chapter 12. So 1 chapter. And again, you don't have a lab that's going to be due next week. Okay. So that lab exam, actually I have that lab exam due to open on Sunday. I may not open it until Monday or something like that, but regardless, I'll let you know when that's open. So just remember that the lab exam, the next lab exam is going to just be on the enzyme lab. All right. So if anyone has questions, let me know. Otherwise, we'll meet again on Tuesday. And that's pretty much it. So we're just moving along. You know, we're almost done, right? I mean, we have chapter 12 and then just 15 and 16 and we're finished. Okay. All right, guys. Take care, guys.
Bye, thank you.
Thanks. What happened here? I got